Welcome to this webcast titled Making Blood from Human Pluripotent Stem Cells Using Laminins and Hypoxia. My name is Rönn and I am a postdoctoral research associate working at the University of Edinburgh, and today I will talk about my research in trying to artificially generate blood cells in the laboratory. I have extensive experience in working with human pluripotent stem cells, originally getting my training at the Salk Institute in California, United States. And I was later involved in establishing Sweden's first ES and IPS core facility located at the Biomedical Center at Lund University. I am also an active member of the International Society for Stem Cell Research, helping to promote the interests of young stem cell researchers as part of the ISSCR Junior Investigator Committee. To better understand the purpose of my research, we need to talk about the type of cell that is critical for our survival, the hematopoietic stem cell, also known as the HSC or the blood stem cell. HSCs are a very rare and sensitive type of blood cells that for the most part can be found in the marrow inside our larger bones. These cells are unique and they are only created during a very brief period of time as our bodies form during embryonic development. They are important because they are responsible for providing the body with approximately 1 trillion new blood cells every day to replace old and broken blood cells. Without the HSC's capacity to both self-renew and to give rise to all the various types of mature blood cells, we would not be able to survive. Since the HSCs are very sensitive, they are often lost during stressful treatment against diseases such as leukemia. To successfully make the patient recover, the introduction of new HSCs is therefore necessary and will usually feature cells donated from a different individual. However, finding a fitting donor to patient match, together with difficulties in securing a large enough amount of cells with good functional quality, can limit the efficiency of treatment. But if we could figure out a way to artificially generate new HSCs in the laboratory, potentially making HSCs using the patient's own cells, we would be able to circumvent all these problems and significantly improve how we treat blood disease. And that is pretty much what I am trying to do. I am trying to find a way to use stem cells that have the capacity to form all the cells and tissues in the body, that is pluripotent stem cells, and instruct them so that they form new HSCs similar to what happens naturally inside the body during our embryonic development. Pluripotent stem cells have already been used to make many different types of cells in the lab, but generating HSCs that are as functional as the ones found inside our bodies have been largely unsuccessful. However, I think I might have found an answer, and it relates to an idea I had earlier in my career when I was a PhD student. In my PhD thesis, I postulated a theory called the model of functional impairment by external oxidative insult. In a nutshell, I proposed that successful generation of HSCs will be fundamentally incompatible with the standardized ways of cell culture that we are currently using in research, and that the key reason why HSCs cannot be generated is due to that they suffer from too much oxidative damage that result from their exposure to atmospheric levels of oxygen that we have when we perform experiments in the lab. As I mentioned, HSCs are very sensitive, and it's already well known that they can only be kept outside the body for a short period of time before they start losing their functionality. And it has been demonstrated that the loss of function that occurs is directly related to an increase in the intracellular amount of damaging reactive oxygen species, also known as ROS. So, formulated in another way, the assumption that HSCs can be functionally maintained at atmospheric oxygen levels much higher than what can be found inside the body might be fundamentally flawed, and that non-physiological culture conditions itself may be responsible for destroying the functionality of any generated HSCs. So I decided to test this idea during my time as a PhD student. And one of the first things I did was to measure the amount of reactive oxygen species in blood progenitors that could be generated in the lab, and to compare these cells to donated cells that had not been exposed to cell culture. I found that almost all of the cells generated in the lab had a very high level of ROS, much higher than what a functional HSC may have. Making this even clearer was when I sorted out my generated HSC-like cells and found that it was only a very small fraction of these cells, the fraction with the lowest available amount of ROS, that had any capacity at all to grow after separation. In the same study, I used various ways to help reducing the amount of ROS in my cells, and I found the most efficient strategy to do this to be simply reducing the amount of oxygen that we expose our cells to while we do cell culture. However, there were technical limitations to how much I could reduce the amount of oxygen at the time before my experiment started failing. And while I could increase the production of HSC-like cells with growth capacity by more than 22 times, I could never really generate any cells that had true HSC functionality or that had a completely normalized level of ROS. So I realized I needed to invent a new way of doing my experiments, a method that would allow me to generate blood cells at oxygen levels so low that they could be considered physiologically relevant, that is, the same as what you would find inside the body. And it is for that reason that I decided to start keeping my cells on laminin. I began introducing laminin-based cultivation as an alternative way to keep and maintain the human pluripotent stem cell lines that we keep in the lab. And here I will give a brief overview how this new routine compared to an older method for cultivating our HPSCs. All that is necessary with the new method is a standard plastic culture plate, some PBS, triple express, a commercially available HPSC media, some laminin 5:1, 15 minutes of my time every 4 to 5 days to do passaging, and of course the 2 to 3 minutes every day needed to perform daily media changes. In comparison, the same cell line is here seen with the previously used culture protocol where the HPSCs are kept as colonies on a supportive layer of murine embryonic fibroblasts, also known as MEFs. This protocol requires preparation of a BFGF supplemented DMEM F12 plus knockout serum replacement based HPSC media and demands careful removal of spontaneous differentiation before the cells can be passaged by mechanical dissociation using a cutting tool. Needless to say, establishing and switching to the laminin-based protocol significantly reduced the workload for maintaining my HPSCs. Furthermore, estimating the combined cost of cultivating a line of HPSCs, I found the new method to cost only a fraction of that of the MEF-based method, and that is not even including the additional cost of acquiring the needed MEF cells. Now, going back to making a method for generating blood cells at physiological oxygen levels, one major necessity was to avoid differentiating my HPSCs in the form of three-dimensional cell aggregates, also known as embroid bodies or EBs. Due to the three-dimensional structure, EBs are by nature very heterogeneous, with cells getting uneven exposure to culture factors depending on whether they are located at the surface or at the center of the EB. This becomes a major problem when trying to cultivate EBs in lower oxygen conditions, since the now critically low oxygen level at the center of the EBs will start to interfere with the ability of the EBs to stabilize and grow. So, to enable cultivation at physiological oxygen levels, I decided to circumvent the use of EBs and instead differentiate my HPSCs as a flat monolayer. This way, all cells get a more uniform exposure to the factors in the culture, giving me better control over the cell's behavior while also avoiding the growth failure associated with EBs. In other words, using laminins to keep my cells as a monolayer allows me to go beyond what is possible with older culture methods in terms of hypoxia. Since my work is still unpublished, I cannot go into too much detail regarding the design of the blood differentiation protocol that I am developing. But described in a simplified way, my new system features a three-step approach where in the first step, a monolayer of HPSCs are instructed to start differentiating into the mesodermal germ layer, which is the early developmental branch of cells that consists of the precursors for tissues such as blood, heart, vasculature, and more. In the second step, the cells are directed to form the vascular precursors of blood known as hemogenic endothelium. And in the third step, factors are added to encourage the formation of blood cells, including cells that look the same as true adult HSCs. To visualize, on the left, you can see pluripotent stem cells shortly before the start of an experiment, and on the right, you can see the large amount of round and non-adherent blood cells that are formed at the end of the experiment. While much additional work is still required, I am confident that developing methods that help protecting the cells from damaging oxidation will be a key step in helping us generate cells that are better and more functionally suited to eventually be used for clinical applications. And perhaps this concept is not limited to the generation of blood, but could also be applied for improving the generation of many other cell types found in the body. With that, I would like to conclude this presentation, but if anyone would like to know more about my research, please feel free to find me through my LinkedIn profile. Thank you very much for your attention, and good luck with your future experiments.